Good morning and good afternoon, everyone. It is my pleasure to welcome you and our speakers, Dr. Paula Littlejohn and Dr. Brett Finlay, at this year Joanna and Brian Lawson Seminar in Microbiome Nutrition Research. My name is Elena Comelli. I'm an associate professor in the Department of Nutritional Sciences at the University of Toronto, and I'm also a scientist at the Joanna and Brian Lawson Center for Child Nutrition. This is a network of researchers and educators affiliated with the University of Toronto and working towards better lifelong health through reducing child malnutrition and chronic disease. I'm really honored to host this seminar together with Chelsea McPherson, whom I would like to thank for all the work in organizing this event. I would also like to thank Joanna and Brian Lawson. Thank you for your vision and for making this event possible. So we have more than 200 people who are registered today from coast to coast, all very much looking forward to the two presentations. So as usual, our two speakers will give their talks first, and then there will be a panel for the Q&A session, which will last about 15 minutes. You can enter your questions in the chat if you wish, or if you prefer, you can ask your questions live during the panel. So, and now without any further ado, it is my pleasure to introduce our first speaker, Dr. Brett Finlay. Dr. Finlay is a professor in the Michael Smith Laboratories at the Department of Biochemistry, Molecular Biology, Microbiology, and Immunology at the University of British Columbia. He received his PhD in biochemistry at the University of Alberta, and then he conducted postdoctoral studies at the Stanford University School of Medicine. He joined UBC in 1989. So research in Dr. Finlay's lab focuses on host microbiome interactions at the molecular level. They really are at the forefront of the field of cellular microbiology. Dr. Finlay has made several discoveries in the area, and he has published more than 543 papers. His laboratory studies several pathogenic bacteria like Salmonella and E. coli, and then more recently the microbiota. He is so well recognized internationally, has won several prestigious awards. This is an impressively long list, so I will mention the CHR Distinguished Investigator Award and the prestigious Canadian Killam Health Sciences Prize. Dr. Finlay is a fellow of the Royal Society of Canada and the Canadian Academy of Health Sciences. He's an officer of the Order of Canada and Order of British Columbia, and inducted into the Canadian Medical Hall of Fame. So Dr. Finlay, thank you for being here with us today, and we are very much looking forward to your talk.
Thank you. Thank you, Elena. Thank you, Chelsea. Let me just see if I can share this. Great. So it's a real pleasure to be here, especially as I get to be the warm-up act for my former PhD student, Paula. And so what the plan today is we're going to tag team it. I'm going to talk about malnutrition in the microbiome and stuff we've done in the lab, both in the mouse and the human models. And then Paula will talk more about some really exciting stuff on multiple micronutrient deficiencies, now how that impacts the microbiome. So just to get started, I would like to acknowledge I'm on the land on which I work is the traditional, ancestral, and unceded territory of the Musqueam people. This is a picture of UBC on their ancestral lands. So, as you know, malnutrition is a huge factor worldwide. It consists of, let me just get this screen out of the way here. It consists of stunting, wasting, and it affects probably about half the children worldwide overall. And Paul will say more about this in the micronutrient deficiencies as well. And it's a major problem, and it's a complex problem. And when we first started working in this area, I thought, well, you just feed the kids, they'll get better. And that turned out not to be true. And there's many, many things involved in it in the environment. Obviously, it's diet, but it's exposure to antibiotics, to fecal contamination. The microbiome is thought to play a role, but we really know very little about mechanistically how this whole thing works. And so several years ago, we got into this, looking at something called environmental enteropathy, or EE. And this is a disease that had been described in malnourished kids for many years, but they knew very little about it. It was characterized by two major things: intestinal inflammation and then blunting of the intestinal villus. You can see in this picture where the villi are quite blunt in the lower panel here. So we knew this existed. We knew this was in kids, but really didn't have very much knowledge about how this all worked. It was thought that, it was recurrent to pathogens that are constantly infecting these children, causing this problem. And we presumed, but didn't have data to show that there were changes occurring in the microbiome, and this is somehow affecting this whole malnutrition sort of phenotype. But as I said, at this time, there was just no animal model. We knew very little other than the odd biopsy taken from children. We saw this happening, but we had no idea what was going on. So a grad student in the lab named Eric Brown decided to tackle this project. He said, well, let's see if we can get a mouse model. So what he did is he basically put mice on either normal controlled diet, which has a fair amount of fat and protein in it and some carbohydrates, or on a diet that more resembles what you would see in underdeveloped countries, which is basically less fat, less protein, and a lot more carbohydrate. That's what they call the regional basic diet. So the idea is, let's put them on a diet similar to what these children would get, and can we basically recapitulate all the symptoms we see in EE. So Eric said about doing that. And when we change the diet, and in panel A, you can see the comparison to diets of the control and the malnourished, or mal, in panel A. And you can see that the malnourished has a lot more carbohydrates and less fat and proteins, but the total calories were still the same. So, okay, that's good. So then we started to look at what happens to these mice. And first thing you look at, of course, is weight. Do they gain weight? And you can see in panel B that the malnourished diet, they gained less weight than control. Okay, so they're starting to be stunted. They're just not growing as well. And you can see weight gain at day 21 in panel C. However, we didn't really see any difference changes in the microvilli. That's in panel G in the bottom right corner there. And we weren't seeing a lot of the inflammation. We did see some intestinal permeability. That's panel F, showing FITC leaking into that through the intestine. But it wasn't recapitulating everything. So basically, we're kind of stunting them, but we're not recapitulating the whole EE disease that we see in these malnourished kids. So we did look at the microbiome, and not surprisingly, it was very different. You can see in here, when you just compare the duodenum or the ileum and the microbiome compositions, they were quite different between malnourished and controls. So yes, there were microbiome differences, and we weren't quite sure what that meant at that time, but we were seeing the changes in the microbiome. But we still didn't really have the model we wanted. So you know that children in these countries are exposed to a large amount of basically fecal coliform counts just in their environment. Water isn't as clean. They're exposed to environmental feces, dirty waters, types like this, and the hygiene is not as great. So we knew they're exposed to this. The other very characteristic of kids with this malnutrition is what we call small intestinal bacterial overgrowth, or it's also called SIBO. So what that means in the large intestine, you're seeing, in the small intestine, you're basically seeing fecal microbes that are characteristic of the large intestine. So the concept is they're ingesting these microbes, they're then getting into the small intestine, and they might be impacting things. And this is obviously due to the fact that they're probably ingesting feces in their environmental exposure. So Eric reasoned, well, could this play a role in could this actually help potentiate the symptoms of what we see in malnutrition? So he set up a very clever experiment where he put the mice on the malnourished diet, but then he orally gavaged in a collection of different microbes. And you can see the collection called BG. This is one I want you to pay attention to because it's one that worked. Basically consists of 6 microbes that are characteristic of some. And then he did a Clostridium group, a Prevotella group, and you can read the others. So he basically put these different groups of microbes, orally gavaged them into these animals, and said, what happens? Do we actually start to recapitulate the environmental enteropathy we see in these children? And so lo and behold, it turns out that one group had a spectacular effect on these things, and that was the BG group. So just to remind you, the BG group is a bunch of bacteroides, basically any coli. So if we look at weight gain in panel A, if you look at the malnourished, you see the red bar, that's the BG group. So they gained way less than the others. The others were stunted in the malnourished compared to the controls, but nowhere near what we saw with the BG group. And the other way you can actually measure sort of growth, which I didn't know until we started these experiments, is you hold a mouse up and measure the length of the tail, and that's really an indication of growth of the whole body. And you can see the BG is heavily diminished in terms of tail length. So, okay, well, we're getting a more effect on the whole stunting, but does this actually look like environmental enteropathy? So it turns out that Eric did all the sections of these mouse intestines, and lo and behold, yes, indeed, it worked beautifully. So if you look at the top left panel where it says mal-BG, so this is a malnourished animal where we gavaged in that group of 6 microbes in the BG group, and look at the villi, you can see how short it is. And compared to all the others, which didn't have this villus blunting. So you can measure the villus blunting, that's what you see in the bottom panel here, and you can see that the BG group had very severe villus blunting. So that was really exciting. Now, the other thing that we wanted to recapitulate was intestinal inflammation. And you can see in the bottom right corner, when you start measuring inflammatory cytokines, that when you add the BG group, we definitely got a very large increase in inflammatory cytokines. So we were very excited then because we felt, aha, we're starting to recapitulate the symptoms that we expect to see in environmental enteropathy. So we then looked at various other parameters that are characteristic of this disease, and one of them is obviously intestinal permeability. And it turns out that, yes, indeed, the BG group caused a major change in intestinal permeability. Probably the best panel to look is panel E, where you're looking at zonulin seeping through the intestine and measured in the serum. And you can see in the bottom right that red bar here, there's lots more permeating through. We also saw changes in expression of tight junction proteins and FITC leaking through. So hooray, we got increased intestinal permeability. We felt this is a key thing of the disease, and it recapitulated there. And then here's a more detailed look at the villus blunting. And again, in pictures on panel F, you see a very big change in the morphology compared to the other 3 groups there. The villus are heavily blunted, and you can measure the height. You can see this decreased height, and also the villus crypt ratio, you can see it's much decreased. So we were very excited about this because we've finally been able to recapitulate what we see in these kids. Again, more details about the inflammation. Calprotectin was highly elevated in these things, as well as the inflammatory cytokines. So we were seeing the inflammation that we'd hoped to see in this model. So to put it all together, that we were very confident that we'd actually, for the first time ever, developed a really good model that we can really study the whole concept of environmental enteropathy in an animal model. So basically, we put them on a diet and expose them to this particular group of microbes, the E. coli bacteroides group, and then they trigger all the symptoms we see in children: intestinal permeability, the dysbiosis in the microbiome, growth stunting, inflammation, and villus blunting. So that was great, and basically this model has now allowed us to explore various other parameters. So now that we had the model, I want to tell you about where another graduate student came in, and this is Linda Bauer. And she wanted to look at the role of malnutrition in the concept of the gut-brain axis. So we know that malnourished children do not neurologically develop to the same extent as those that are well-nourished. We know it impairs their cognition and many other learning abilities and things. And we know the whole gut-brain concept is there. Microbes influence brain development. We know germ-free animals have basically less developed brains and things. But we want to see, what is the impact of malnutrition on their cognitive development using this animal model? So Linda started on this, and one of the areas she looked at is, in the story I'm going to tell you, is the microglia. So microglia are cells in the brain that are basically kind of like macrophages. They're called the macrophages of the brain, really. One of their jobs is scavenging for pathogens that may make it into the brain. But the other job is they clean up the brain. They remove dying neurons. And importantly, they're involved in synaptic pruning. So this is basically needed in order to learn and remember things. And so they play a major role in learning and development too. And the reason Linda wanted to have a look at this is because a paper came out just when we were starting this, where they looked at the microglia in the brain of normal, and this is FPF mice versus germ-free mice. And if you look at the panel F, you can see the SPF mice, that's what the microglia look like. The germ-free look completely hairball different. You can see that they have basically much more, basically these spidery things coming out from them. They have number of points, number of segments. They're much more branched out, shall we say, and they look really, really different. They also could recapitulate this phenotype if you took a normal mouse in panel D, the SPF, and then you treated it with antibiotics. This too affected the brain development of these microglia. And if you took germ-free and then put in normal microbes back into it, you could actually get rid of the symptoms. They appear much more normal than the germ-free in the top right corner. So we wanted to ask what happens to microglia in the brain of these things, and could that influence mouse behavior? So Kailinda had a look at this, and basically what she saw was very spectacular. So again, to remind you, in the EE group, that's one where we do the mal plus the Bacteroides group, BG group, you can see that the microglia look very, very different compared to just the malnourished. And the malnourished even look different than the control. So we're obviously impacting on brain development of these things in the malnourished and the EE group. And so that's when we started to have a look, well, what's going on in terms of brain and learning. So here's a very pretty picture that Kailinda put together, just looking at microglia in the control and malnourished. You can see there's major changes, and there's major increases in what are called endpoints and branch points. And really, it says these microglia are very different in the malnourished and the E group than they are in the control group. Okay, but great. Well, what does that mean? Does it affect the mouse's ability to do anything? And so then Kailinda started down this long, torturous journey, shall we say, of looking at how these mice behave and looking at various behavioral studies. And one of the one is what's called an open field test. And so Kailinda went to Walmart and got a great big Tupperware container and put little tape marks on it. And then you put a mouse in there and you ask, what does a mouse do? And you really measure how much time it spends around the edges versus in the middle. Now, a normal mouse is shy and does not want to be out in the open areas, and it very rarely goes through the middle of this thing. And so if you see differences in mice in where they go in these things, you then can get an idea of their behavior. So Kailinda spent many, many days and nights in the mouse house with a GoPro over top this makeshift open field test measuring these mice. But it all worked out, because basically what they found is the EE mice, for example, they spend a lot more time in the open field, which means this is affecting their behavior, and they're more adventurous, which is not good from a mouse's point of view. So here you can see some traces. You can see the EE group spends a lot more time going through the middle than, say, the control, which really generally tries to avoid it when you look at these traces of where these mice go as they spend their time in this open field test. So another one she did is what's called a Morris water mace, where you basically put a mouse in a pool with clouded water, and it's got a platform in it, and it then is able to swim to that platform and find it. But then you move the platform, and you basically time how long it takes the mouse to find the new location. And what happens if learning is impaired, sometimes they basically can never find it, or they just don't remember it. If it's not there, they basically freeze, and they just basically drop to the bottom of the pool, and then Kailinda has to fish them out. So you can see where it says probe 2, bottom right corner, you can see that in the malnourished, there was some freezing, but it was even more prevalent in the E group. So this is really affecting their ability to learn where things are and their spatial memory and how it all works and their cognitive plasticity. And we can see that both malnourished and especially the E group had profound effects on the mouse's ability to do this, as measured by their freezing ability. So Kailinda compiled all these things here, and you can see the various tests that she did in this. The other one we saw a big effect in is what's called a novel object recognition test on the left-hand side. And basically, this is where you put, say, a marble or some other object into a mouse cage, and you measure its ability. It's basically curiosity to go and explore it and see what's going on. And you can see that that the E group in this test is very, very different than the control group. It's really affected their ability to probe these novel objects and their curiosity. Some other tests didn't have effect. Light dark test had no effect, which measures anxiety. And I've already talked about the Morris water phase. So now Kailinda also then stumbled into something else that we didn't really quite expect, but it turned out to be a real fascinating story. And she found that the livers in these animals were very different than the control group. And basically, she did a whole bunch of stuff. I'm not going to go into a lot of detail here. You can read about in her paper. But basically, she found there were very, very different looking livers in these things. And when you look closely, the liver triglycerides were extremely elevated in these things. And basically, this basically recapitulated all the symptoms that you see in what's called non-alcoholic fatty liver disease. And this is basically a large metabolic disease you see often in obesity in the liver. Yet these are obviously not obese mice, rather they're malnourished, but it's obviously causing some severe metabolic disease in these mice when you malnourish them and really impacting the liver. Okay, so that's great. It's the mouse model, allows us to probe some of the effects of malnutrition and environmentalopathy, but what about kids? So this is a study we got involved in, and our job was to do the microbiome of this, something called Afrobiota. And just to remind you in terms of kids, is the bulk of the kids that are really stunted live in sub-Saharan Africa and Southeast Asia. Here's a global map. And this study was set up in two places, Central African Republic, the orange arrow, and Madagascar, the blue arrow. And these two countries, there's a phenomenal number of kids that are stunted. You can see in the bottom, a little over 40% of these kids are severely stunted in these countries. So this is a project that's set up by Philip Sansonetti and Pascal Vanache at the Institut Pasteur. Here's Pascal up here. And they set up a whole bunch of arms to the various studies, looking at many, many different things of what's going on in terms of the microbiome, which we did. We also helped with immunology. It's one of our grad students, Kelsey, here, looking at various immunological markers and various other aspects, all looking at these children. So they had just under 1,000 kids all told. And as I said, they were from those two countries in Africa. And they're really broken into three groups. Those are severely stunted, moderately stunted, and non-stunted. And so the question is, what's going on in these kids and what's actually happening? So remember, I told you that small intestinal bacterial overgrowth, or SIBO, is a very hallmark of these malnourished children. And so one of the things this group did, and we were not involved in this, but they basically showed very clearly that these things had a strong, severe intestinal bacterial overgrowth. Remember, these are the large fecal type microbes contaminating the small intestine. And you can see that that's it. This is the next one. The large extent of SIBO. So we then started to characterize what were the microbes in this small intestinal bacterial overgrowth. And the strangest thing is they were actually oropharyngeal microbes. So these are microbes up in the upper respiratory tract, yet we're seeing them way down in throughout the intestinal tract. And we then characterize this, and it was the same in both countries, despite their physical distances from things. So there was a real, we were surprised at this. We thought we would see more different microbes in this. We didn't expect to see the oropharyngeal ones. So we dug into this and characterized the microbes in great detail. And as you know, in a normal person, the microbes are basically compartmentalized to the oropharyngeal. There's very few in the stomach, a few, the duodenum, small intestine, and then large intestine and feces, which is a very different group of microbes. Yet in the stunting kids, we found that these basically compartmentalization was gone. It was really, there wasn't even a gradient of microbes. We were seeing the oropharyngeal microbes throughout the intestinal tract, way down in the feces, et cetera, and the stomach and duodenum too. So you can see in the far right, we kind of summarize what we're seeing here. We're seeing the oropharyngeals here, as well as the enteropathogens in both of these things. So it's like the whole intestinal tract has just lost its various compartmentalizations, which surprised us extremely, but seems to be a major contribution to what one sees in these stunted children. So to basically sum up, basically what I've tried to convince you of is that we successfully developed a mouse model that allowed us to mimic the various features that we see in environmental enteropathy. And the key thing about that is it's not just diet. You actually have to gavage in fecal type microbes, E. coli and bacteroides, and these two together, and you can't uncouple them. You have to put these groups together. These two groups seem to trigger all the things we see in environmental enteropathy that we knew to see clinically. The other thing I think that's very important to point out is that malnutrition has a profound effects on not just growth, it affects brain development. I showed you the behavioral studies in the mice. And very surprisingly, it actually resembles metabolic disease, non-alcoholic fatty liver disease, which is very characteristic it's very characteristic of obese type people. And we were very surprised to see them malnourished, but they almost seem like they're basically the opposite sides of the same spectrum where you see this disease infecting the liver. And then in the children, we saw that, yes, indeed, stunted children have a very large small intestinal bacterial overgrowth, but you also have uncoupling of the entire GI tract, And you also have oropharyngeal microbes throughout the whole GI tract. So this is a very bizarre microbiome type effect that we're seeing, where you're seeing microbes from the mouth through the intestinal tract and things, which I presume contributes to the whole disease phenotype we're seeing in these kids. So the work I showed you was mainly 2 graduate students, Eric Brown, who did the malnutrition model, and Kylinda Bauer, who did the behavioral stuff. And then we collaborated with the Pasteur group, Pascal and Philippe, to develop the whole Afrobiota microbiome characterization of these children. And Christophe Borsch was involved in metabolomics. So with that, I will say thank you and stay tuned, because Paula's got some really cool stuff coming up on multiple micronutrient deficiencies. So let me stop sharing.
Thank you.
Great.
Thank you, Dr. Finlay, for this fantastic talk. I'm sure there will be lots of discussion at the end, a Q&A session. And but now, yes, let's go straight into the talk of Dr. Littlejohn. So Dr. Littlejohn currently is a postdoctoral research fellow, co-supervised by Drs. Cobor and Valens at the University of British Columbia and the BC Children's Research Institute. Dr. Littlejohn received her PhD in microbiology and immunology from UBC in the lab of Dr. Finlay. And here she developed the first animal model to examine the impact of postnatal exposure to multiple micronutrient deficiencies on the gut microbiome and the gut resistome. Currently, her work focuses on understanding the role of maternal and postnatal nutrition in modulating epigenetic changes that have long-lasting effects. We will keep this for a future talk. And for now, we go back to your PhD work, Dr. Littlejohn. Please, you can go ahead.
Thank you, and for the very warm welcome and the invitation. Okay, and thank you, Brett, for such a wonderful story. And so I'm going to show you just how I built on the great work that was done before me and stand on their shoulders to propel myself into my PhD. So specifically, what I'm going to talk about today is the role of micronutrient deficiencies in early life, and specifically focus on the gut resistome and antibiotic resistance. And so, as Brett mentioned, there seems to be a microbial contribution to malnutrition that has been well characterized. And so when we look at it, we've seen it in overnutrition. When you think about malnutrition across the spectrum, it includes both undernutrition and overnutrition. And then we've seen, we've done sequencing to understand this, and we've looked at both humans, and we've looked at different animal models such as mice and pigs, piglets specifically, and we've also looked at children. And essentially, we find some of the same things, and we look at different diets. So we've examined protein deficient diet, we examined the regional based diets, and then zinc diets. Actually, let me move this screen here. I looked at zinc diets as well in malnutrition. And basically what we've and others have found is that when you look across malnutrition, whether it's overnutrition or undernutrition, we kind of see some of the same signatures. So we see an altered microbiome, both in composition and diversity. We see expansion and a loss of certain species. We see an immature microbiota for age specifically, and we look at the undernourished group. And then we see some functional, biological, and clinical phenotype. And then this can be, like Brett mentioned, can be even behavioral tests. And then it can also be just some of the metabolic features that we see, and then just how the microbiome actually performs in general. But what hasn't been looked at is the role of micronutrient deficiency, specifically as multiple micronutrient deficiency on the gut microbiome. And so what exactly are micronutrients? Many of you probably already noticed the Nutrition Center, but just for those who don't. So micronutrients are vitamins and minerals that our bodies need in small quantities. They perform indispensable roles. They act as cofactors, immune function. They participate in immune function. Of course, our vision cycle, everyone knows about vitamin A. They play a role in DNA synthesis, you know very well with the B vitamins like folate and B12. They also participate in gene regulation and so much more. And approximately 30 micronutrients are needed daily for optimal health, which I can tell you most of us are not achieving, including myself. But it turns out that a huge part of the population actually don't achieve this amount. And so indeed, micronutrient deficiency, also known as hidden hunger, affects 2 billion people globally. Of this, 340 million are children under the age of 5, which approximates to about half of all children within this age range globally. But not all micronutrients actually contribute to micronutrient deficiency. And the main culprits that have been well studied are vitamin A, iron, zinc, B9, also known as folate, B12, cobalamin, and iodine. And then it turns out that while vulnerabilities to micronutrient deficiency remains throughout the life course, it appears that those that occur during this early window, this first 1,000 days of life, confers both immediate and lifelong consequences. And so my work has been focusing on that, and a lot of others have also focused on trying to understand what happens to the gut microbiome, as well as the body in general in this first 1,000 days when it's exposed to micronutrient deficiencies. So just a quick overview of what the human microbiome is. It includes both archaea, bacteria, fungi, viruses, and parasites. And this together all comprise of our microbiome. And then we have them at various sites. Brett mentioned about this oral microbiome that can then perfuse down into the small intestine and the large intestine in malnourished children. And then we have your genital microbiome, skin microbiome, and then our digestive tract microbiome, which is the main focus of our work in the lab. And so using this, but one of the things that's not well characterized is what's known as the gut resistome. And many of you may or may not have heard of this. And so the resistome is the collection of genes or genetic material that confers antibiotic resistance. And it turns out that resistance genes were found in 30,000-year-old permafrost, and also in remote societies such as the Hadza tribes and Amerindian societies. So suggesting that these antibiotic resistant genes didn't just come from our introduction of antibiotics into population. They existed, they predate our antibiotics. And so they already existed. And this is a mechanism that bacteria use to compete for resources, to compete for real estate, especially in the gut microbiome or even in the soil. And so this has been well documented, especially in the environmental fields. Within the gut microbiome, it is the largest reservoir of antibiotic resistance, actually lie with the commensal bacteria, which seems a little counterintuitive. Of course, we think antibiotic resistance. We always hear about the antibiotic resistant bacteria, and they're often pathogens. But when you think about it, just in sheer numbers, the commensal bacteria, there's just so much more of them. And so they just have more of these genes that they can lovingly pass around. The resistome is also present at birth, but it also seems to reduce over time as the gut microbiome matures, and is shared among community members through horizontal gene transfer, and can also be carried by mobile genetic elements, which I'll get more into in later slides. So how does resistance happen? And so when we think about resistance in our context, looking at antibiotics, so if you have some bacteria, some of them may just naturally have resistant genes, as I mentioned, with the 30,000-year-old permafrost, so they already exist. But then when you introduce or expose them to antibiotics, you'll get some killing of some of the microbes, but the ones that have these resistant genes, they'll stay alive. And then what they'll do is just happily pass it on to their friends within the community, and then they'll continue to grow. And then they share these, and then now the entire community has these resistant genes. And so this is basically a simple explanation of how resistance actually happens. But then how do they get disseminated? And so they can get disseminated by what's known as these mobile genetic elements. And so just as it says, they're mobile, they can move around, they carry genetic material, and they can impose those into different bacteria. And so plasmids, the circular DNA that can move between cells. We can have these transposons that can actually insert into the chromosome, and they carry the genes that are also resistant. And then it can also happen by bacteriophages. And so these phages can then, these are viruses that can attack bacteria. They carry DNA, so they can pick it up from one bacteria that might be antibiotic resistance, and then they pass it on to the other bacteria as they infect them. And then they do this in several different ways. They can do this by transduction. And so basically they just pass the genes on to the next cell. There's a donor cell and a recipient cell. Transformation and then transduction, where the bacteriophage can actually enter and carry that antibiotic gene with it. And then it can also happen from conjugation. And so this is basically, your bacteria having sex, basically. So once the cell come in close contact, and they can share that resistance mechanism to the recipient bacteria. And I don't think I need a lot of to explain or to say or convincing really to say that antibiotic resistance poses a big threat to global health. It is a growing crisis worldwide. We don't have enough resources to even combat this. We need more surveillance. We need more judicious use of antibiotics. And so we really do need to understand how resistance actually emerge within different populations. And then we'll be able to think of different ways that we can mitigate some of these effects. And if we look at this recent data that was a paper that was published, I believe this was the Lancet, and it mentioned that 1.2 million people died from drug-resistant infections in 2019. So this is really huge. However, even knowing all this, the gut resistome is still understudied, especially in malnutrition and more specifically in micronutrient deficiency, and definitely when we look at multiple micronutrient deficiencies. And so based on Eric's project and just looking at the micronutrient deficiency, I went on to develop this mouse model to both examine the gut microbiome and some other biological phenotypes I was interested in. But this model, basically, we took 3-week-old black 6N mice. Then we put them on a control diet or a low micronutrient diet. And so the diets were, as Brett mentioned earlier, the same as Eric's model. So they were isocholeric. So, but in this case, there wasn't any protein deficiency or fat deficiency. The fat, protein, and carbohydrates were exactly the same. The only difference between these two diets were the micronutrients. And I chose 5. And these are 5 that are focused on by the Micronutrient Forum as a global malnutrition issue that they're addressing. And so I looked at that. The other reason why I didn't choose iodine is because iodine is almost on its way out of being eradicated, with about 70% of the world population right now has access to table salt. And so I decided not to do iodine with this. And so we see here we have iron, at just 5 milligrams in these mice compared to 35 in the control. Vitamin A, pretty much 0, vitamin 12, 0, B9, 0, and in zinc, very low zinc, from 27 milligrams in the control mice to 2 milligrams in these low micronutrient mice. And then also I want to mention that this model was also infection-free and antibiotic naive, and you'll understand why as I continue. So just looking at some of the features like Brett mentioned earlier, so one of the things we always want to examine, well, first, sorry, I wanted to make sure that we depleted the micronutrients in these groups. And so I show here that, yes, we depleted all the micronutrients that I set out to do in, not to the same degree in different tissues and serum, but we definitely saw that the model did work. We were able to deplete the mice of these micronutrients that we set out to do. And then from here, we measured weight, which is something that we just typically look at when we look at our malnutrition models. And then you can see here, these mice actually went out to day 28 and not day 21. So I extended Eric's model a little bit by just a week. And we saw that there's a clear separation between the low micronutrient group and the control group. So there's significant growth faltering that happens starting at week 2 in the mice fed the low micronutrient diet. And then when we quantify their stunting, we use a tail length as Eric did in his previous model, but I also added tibia length. And I added tibia length for several reasons. One, shorter leg length is also associated with increased risk of obesity and metabolic disease later in life. And I wanted to kind of keep that in the background as I continue to investigate this model. And then I also want to mention that there was no significant difference between the amount of chow consumed. So these mice weren't actually starving. They were just eating the appropriate amount for their body weight. So you see a slight difference here, but it's because mice consume actually 3 to 5 kilogram per body weight of chow. And so these mice are smaller, so they're obviously going to eat less, but there was no significant difference between them, and these mice were not actually starving. We then went on to characterize the gut microbiome. And if you can just look here visually, you can see, so this is day 0, and then at the end of the experiment, day 28, and this is the control group. And so what we'd expect to see is a more balance of the microbiome as it matures. And so we'll see more the balance between Bacteroides and Firmicutes to be almost equal, which is what we will see in a typical microbiome. We don't expect to see proteobacteria. They start at the beginning as they begin to colonize and make the microbiome more habitable for other species to come in, but then they sort of disappear as the microbiome matures. However, in the low micronutrient group, you can see that the microbiome looks almost exactly the same, as if nothing changed. The only difference was this expansion of proteobacteria. And so these are the bad bugs that you don't want in your gut. So the E. coli, Klebsiella, and so forth. But this is metagenomic sequencing, and so we wanted to understand, well, is there actual biological readout? Can we actually quantify this biologically? And so we took fecal samples from these mice, we planted them on McConkey agar, and then we quantified or counted the CFU to look at Enterobacteriaceae, and we saw that yes, there was significantly more Enterobacteriaceae in the mice with the low micronutrient group compared to the control group. And again, no infection, no antibiotics, just being starved of micronutrients. These mice actually had more Enterobacteriaceae. When we looked at different species in the microbiome, we saw increase in Klebsiella, we saw increase in E. coli, Haemophilus, but we saw a decrease in Streptococcus and Staphylococcus, as well as Prevotella. And just want you to really just remember, especially these three, because I'll come back to them later, and then Strep and Staph. So then now I moved into examining the resistome. And the reason why I did this is because I did a previous metagenomic analysis. I saw some significant differences that were really exciting in looking at virulence. And I also saw, some things going on with antibiotic resistance. I didn't know too much about antibiotic resistance, but I was like, well, this is really interesting. I should at least explore it and see what's going on. So it turned out that there's certain 5 mechanisms which bacteria actually gain resistance. So if you think of this as being a bacteria, they can alter their membrane permeability to actually prevent the bacteria from coming in, so they can downregulate these viral structures known as porins so that the antibiotics can't actually get in. They can produce these antibiotic resistant genes, which is mostly what I'll be talking about today. And then they can also upregulate efflux pumps, and these efflux pumps basically work as the bacteria comes in, the antibiotic actually comes into the bacteria, it just constantly pumps them back out. And so this is another mechanism that resistance can actually happen in bacteria. They can also produce these enzymes that can actually inactivate or degrade the antibiotic even before or when it enters into the bacteria. And so this actually is very effective, and I'll talk about some ways that we saw in this model. And they can also alter the target site so that the antibiotics can't actually bind, and by using these different proteins and enzymes to protect from antibiotic binding and killing. So when we examined each of these specifically, looking at altered membrane permeability, we saw an increase in lysozyme, and this is actually produced by the Panna cells, and is also in our bodies. That can actually, it's a natural resistance mechanism, and it's an actual, it can actually kill bacteria. And so we saw resistance to lysozyme, and then we saw this mostly in Klebsiella. We looked at efflux pumps. There were several of them, but I'm just showing the one that most people are probably more familiar with. We saw that a significant increase in genes for multi-drug resistant efflux pumps. We looked at target site modification. We saw these tetracycline resistant protein, this protein M. And so this is a ribosomal protection protein that actually functions to prevent antibiotic action. And then we looked at some of the enzymes, and one in particular, beta-lactamase. And so this enzyme actually inactivates or degrades beta-lactam antibiotics. And so we saw a significant increase in the gut of the low micronutrient group. So showing that just having these low micronutrients, we saw all 5 of these mechanisms were actually induced. And then when we looked at total antibiotic resistance, we saw that that was significantly higher in the low micronutrient group compared to the control group. And these are all a combination of the efflux pumps as well as all the different antibiotic resistant genes that I will get to talk a little bit about today. So some of these resistant genes are vancomycin. We saw resistance to at least gene capacity for vancomycin resistance, also methicillin and fosfomycin, which is a very old type of antibiotics, but it seems to be coming back into use. And then, but we didn't see it across the board. And so we didn't see any resistance to fluoroquinolone, suggesting that these mice then remain susceptible to fluoroquinolone. And so this could potentially be maybe an antibiotic that can be used in micronutrient deficient children, because currently we don't see that there is any resistance in a resistance reservoir that is actually there in low micronutrient deficiency. And so potentially this might be something that will work. And then also in erythromycin, we didn't see a difference between the group. So then Brett had this wonderful idea of like, well, Paula, that's just genes. He was looking at metagenomics. Can you see a biological phenotype? And so we took the fecal samples from the mice, And then we plated them and let them grow overnight, exposing them to antibiotics on each disc. And so this is vancomycin, tetracycline, and methicillin. And so you can see in the control group, this really nice zone area here showing that the vancomycin actually killed the bacteria. And then if you use like a sterile loop and even scraping that area, there's nothing there, it's just agar. And so the bacteria were not able to grow anywhere close to this disc within this zone. However, when you look at the low micronutrient group, what was really interesting is that, if you remember the first slide that I told you, that antibiotic, how resistance happen is that you have some bugs that have resistance and then you expose them to the antibiotics and then you get the whole community, it might kill some and then you have like a resurgence of it. So what we saw was actually what happened here. So if you can see very faint zone in the background, the vancomycin initially cleared that area, but then the microbes actually grew back. And so I took a sterile loop and then just scraped from any direction of from this disc, and you just had a nice, really nice lawn of bacteria that was there. And these are resistant bacteria. We didn't see any resistance in tetracycline, although it was about the same as the control group. And then with methicillin, there was definitely nothing there. And so we just saw that there was the bacteria just grew right up to the disc and they were resistant to methicillin. So just showing that even without these mice being exposed to antibiotics, and we just exposed them in an in vitro setting through antibiotics on these discs, we saw that the resistant genes that we saw in the metagenome actually conferred a biological readout and a biological phenotype in these mice. And I'm just showing a sample of the size. And then we also had, I also looked at about 6 fluoroquinolones and also saw that there was no resistance. So if you remember the slide that I showed you with the metagenomics analysis, that these mice might be still susceptible to fluoroquinolone antibiotics, and we did see that biologically. And so from this, we conclude that our metagenomics accurately predicted some, but not all resistance. And so one of the questions I often get asked is like, well, what could be triggering this resistance? They're not being exposed to antibiotics. Why are these microbes behaving like this? And so one of the thoughts in the literature is is oxidative stress. And so oxidative stress can actually induce microbiome to actually do just very different things. They can produce ROS to actually self-destruct. And then if they have resistant genes, bacteria in the area will just pick that up, as I talked about earlier. And so then they become resistant. They can produce these ROS to actually kill off other bacteria. But what we saw in this mice was that there was significant oxidative stress in the mice in the low micronutrient diet, but the response and the ROS protection was about the same, and the stress response. So these are the alarm hormones that are being triggered. So the SOX-R genes to be able to detect that stress is happening were higher in the low micronutrient group. And then we saw that regulation, these genes were actually higher in this low micronutrient group, suggesting that they're trying to deal with the oxidative stress, but unable to. And so we believe that this stress of just not having these nutrients are causing these microbes to then produce these antibiotic resistant genes. And then one of the things that we wanted to examine was like, well, how is it being disseminated? And so there's several ways that antibiotic resistance mechanisms can actually be passed on through bacteria. I had mentioned earlier bacteroidetes. And so this is actually the group that has been well characterized to carry the largest reservoir of antibiotic resistance. And specifically these conjugative transposons, so this is a mobile genetic element, actually is known and characterized for transmitting or disseminating antibiotic resistance. And so we saw in all of the proteins that we looked at that there was significantly higher in the low micronutrient group, suggesting that there's an increase in horizontal gene transfer. So meaning that the community not only has a forming reservoir, but now they're sharing it with all the bacteria that are in the environment. And then we also examined 2 main classes of integrons. And so these are enzymes that are altered, or completely their role is just antibiotic resistance dissemination. And we saw especially class 1 integrons that was significantly higher in the low micronutrient group, basically at 0 in the control group. And then class 2 integrons, again, also higher, suggesting that there is this transfer that is happening between the microbiome. And then just circling back to what I talked about earlier about antibiotic resistance and just the huge impact that it has on society. When you look at the specific bacteria that contribute or that are associated with antibiotic resistance. We see E. coli, we see Staphylococcus, Klebsiella, Streptococcus, Cyanobacteria, and Pseudomonas. When you look at the microbiome in the low micronutrient group, we saw that 3 out of the 4 are actually, 2 here are higher, and there's some others I didn't show here. And E. coli and Klebsiella are higher in the low micronutrient group. Staphylococcus was lower and Streptococcus lower, but suggesting that because they're in this community, there could be some sharing going on. And so the reason why I bring this slide up is just to show that if you already have this reservoir of antibiotic resistance within the commensal bacteria, and then now the microbiome or the diet has induced these pathobionts like E. coli and Klebsiella, then there's more of a chance of resistance being passed on. And this may actually explain why we see these bugs actually appear and why we might see them more in malnourished children. It's because the diet itself is actually first creating that community, first of all, to actually exist, and so creating their abundance. And then the bacteroides are actually now sharing those resistant genes to them as they enter in. And so that's what we're seeing. That might explain what's happening. So in summary, just wanted to share that postnatal exposure to multiple micronutrient deficiency causes growth failure and stunting, alters the gut microbiome and the resistome. We show for the first time a role of multiple micronutrient deficiency in inducing clinically relevant antibiotic resistance, which is a global public health threat, and that metagenomic sequencing may have some utility in predicting biological phenotypes. Whether or not this can be transferred to the clinic will need some tweaking, but it actually could be used in future studies to be able to begin to tease out which child might have resistance and then tailor the antibiotics appropriately. And one of the things I think for me is also a big takeaway is I'm not in any way saying that we shouldn't give antibiotics to malnourished children. But what the research does show is that antibiotic is being used prophylactically and given to kids with uncomplicated malnutrition. And that's where the problem lies, because if they don't need it, then we shouldn't give it, especially from what my data is showing. If there's already this reservoir that exists, then we don't want to compound that by adding antibiotics, which is then going to create more superbugs in the microbiome. So with that, I want to acknowledge my postdoc lab, Dr. Mike Kober and Bruce Balance, and thank you all for your attention today. And I hope you have some good questions.
Thank you, Dr. Littlejohn, for a fantastic talk. Really looking forward to the Q&A. So as a reminder, you can either type your question in the chat, or you can also use the reactions on Zoom and ask your question live. I cannot see all of you, so you can just pick up if I miss your raised hand, and no issues with that. So while we wait for the first question, maybe I just ask one for Dr. Finlay. So in one of the first studies that you presented, I think it was the Brown study, where you used a different mixture of different microorganisms. I think one of those was a probiotic mixture. And it contains plantarum, if I remember from your from the slide. And did you have any specific expectation for that mixture, given the research on probiotic sustaining growth in models of malnutrition in mice?
I think Eric was just grabbing bunches of variety of things. As well, let's try a probiotic and see what happens. As you saw, it had no effect.
Yeah.
Most bacteria had no effect, and for whatever peculiar reason, it was just this mixture of Bacteroides and E. coli that had it. So we were just trying random things. I think Eric just went through the freezer. What could we throw in? And that was one of the things. Let's try a probiotic. Like I said before, you couldn't just add E. coli or Bacteroides. The two have to be together, and they symbiotically feed off each other to allow establishment. But we saw no effect in the probiotics at all.
Interesting. Thank you. We have a question in the chat about SIBO, and if there is any way to prevent SIBO in children, and any specific dietary components, or maybe probiotics or prebiotics.
Well, I'll take a stab at it, Paula. You can help me out if you got any ideas. I mean, obviously it's a big issue, and I think we all think it occurs just because of environmental exposure to these fecal type things, and the kids for some reason are more susceptible to this contamination, and then it seems to set up. It's a terrific question, and I really do not have a good answer. I don't think there's any way that we know of. I mean, the Afrobiota is now trying to see if they can't brush these kids' teeth and things and just alter the oral microbiota that might affect, again, being taken down further into the body. Philip Sansonetti is working on a big teeth brushing campaign in these kids to see if this influences that. But the whole SIBO, I think it's just an environmental impact. They're exposed to feces, and it's just part of the whole picture. Paula, you want to add to that?
Yeah, definitely. I think you brought up a great point there, that it's not just, when we think about SIBO, sometimes we think about SIBO here in our westernized societies and like, oh yeah, just change our diet or do certain things and then we might be able to have some effect. But when you think about SIBO in a community where there's constant fecal contamination in the water, just not having potable water, just close contact with animals, that it's not as easy to actually go in and change the gut microbiome. And you see with nutrition studies a lot, even we can't even change sometimes some of the micronutrients, or we can't reverse stunting. So it's very complicated. We don't have an answer yet, but it is something that people are working on. How can we just target the microbiome? How can we target the small intestine and to be able to stimulate different peptides, that are being produced and be able to shift it. But it's not a simple fix with just a diet. And we have to begin to tailor and try different diets. Some polyphenols might work as well, but we have to fully understand how they're being metabolized and how these microbes are actually using them and what are the end product.
Thank you. Thank you. We have a question on the impact of malnutrition on host physiology, and any thoughts on what comes first? Is it degraded immune versus gut epithelial function?
Classic chicken and egg question. I think the answer is both. I mean, we know that the early life microbiome has a profound influence on how the immune system develops. We also know the early microbiome has a profound influence on gut permeability. So, if these imbalanced microbiome as they're developing, you are screwing up both the gut and the immune system right then and there. So I think they go together. I think it's virtually impossible to uncouple those, and it's just this whole part of the developmental process that gets screwed up. So I don't think you can say, well, they have a screwed up immune system. This allows the gut permeability. It's a bit of both. Paula, you got words on that?
Yeah, no, definitely. It is definitely a vicious cycle. And if you're thinking about the starting microbiome and how the microbiome develops, and it trains the immune system. And so if the microbiome is already, perturbed or altered, and can be from the moms, during, development or during delivery, and so they're going to get certain bacteria from their mom. So if it's already perturbed, and then that perturbed environment is now training the immune system, that's going to be a immune system that's just in a heightened alert state, where it's just responding all the time to inflammation and something that's not right. So I think it is a vicious cycle, and being able to find out which goes first is going to be very challenging. You could probably do some models to look at that. So yeah, in vitro models might be able to give us some highlights a little bit, but yeah, I think it's a vicious cycle.
Yeah, thank you. Do you think any recommendations are possible to improve microbiome health in children to promote their development and growth?
I think with this. So there's a group that did a study recently. It was published, I think, in 2021, and it looked at this component called the microbiome directed complementary food, and they basically put it up against this ready-to-use therapeutic food. And so they looked at creating a food product that's based on local ingredients, so that it's actually sustainable. But the idea was to target the microbiome. And so what they saw was like there were some changes in linear growth in these children. They saw increase in like IGF-1, growth hormones, some of the bone morphogenic proteins. They're also upregulated. Actually, they look at protein. They did a proteomics and look at the blood. And then they also saw some changes in central nervous system genes or proteins. And so There is some work that's being done to target the microbiome specifically with food that are tailored towards the microbiome. And then we are seeing some changes. And there's a couple of studies that are happening now in South Africa as well.
I would add there's other ways too. For example, we've shown that decreased use in antibiotics early in life has a profound influence on, say, asthma development. So that's not a nutrition, but I would say, prudent antibiotic use early in life is critical. And another area that, again, in our asthma work, we're showing that there's certainly some early life microbes that need to be present to inhibit asthma coming along. And so there's talk about, using 3-month-old feces, we can tell whether the kid is heading toward asthma or not. Because of the microbe composition. So there's a look at adding these as a sort of a probiotic mixture to these kids to supplement it. And even things like adding HMOs, high molecular oligosaccharides, milk oligosaccharides to formula, for example. This has a profound influence on the early microbiome, preventing asthma and things. So I think there's a lot more. And then, of course, food, early life food is really breast milk and early life kind of thing. So there's not a lot you can do with that. But I think formula and antibiotics, and maybe there's even discussion of having probiotics that you could also give to these early life infants to influence it. So it's an exciting area, but it's early days. And I think we're still moving in that direction.
Thank you. So did your study show which specific micronutrients deficiency caused more bacterial overgrowth or changes to the microbiome?
So no, I didn't look specifically at single micronutrients, just for that reason, because that's already been done. I wanted to understand multiple micronutrient deficiency and the interaction. And because when you think about children in low to mid income countries, they are often faced with multiple micronutrient deficiencies and not single. And so I wanted to have a model that could best recapitulate what would happen in a real life situation. And what was really interesting about this model is that even with just the micronutrients that I depleted, we actually saw there were some other micronutrients that we didn't intend to deplete, like copper and selenium, that were actually significantly lower in the mice. So they were also depleted, even though I didn't deplete them in the diet, showing that there is some interactions between micronutrients that we don't yet fully understand, because currently what we understand about copper and zinc relationship, at least, is that if you have too much copper or too much zinc, then they'll cause reduction in the other. But here we have a scenario where we have both deficiencies in copper and zinc, even though we didn't even intentionally set out to deplete copper. So I think it's really critical that we understand this nutrient-nutrient interaction versus just looking at single micronutrients. But it's a good question, one that gets asked by everyone who studies multiple micronutrient deficiency.
Thank you. There is a question for Dr. Finlay and about the study where you showed the ability to reverse the spider-like microglia phenotype in the specific pathogen-free mice when treated. Does this imply that the abnormal brain function can be possibly treated with a properly nourished microbiota or other strategies like fecal transplantation?
Because of that, so the brain takes a long time to develop. It's much longer development process than most of the other processes. So, a kid at the first 5-6 years is really brain development, whereas all the other stuff happens much faster. We didn't look that closely at, I don't know, to be honest. I can add that metabolic disease, the NAFLD, that actually can be reversed if it's done fairly early. You can correct that. But we don't really have good answers on whether the brain behavior can actually be changed or not at early times. So we didn't do those experiments. It's hopeful, but I guess the idea would be to try and prevent it or turn it around as quick as you possibly can. That, as Paula pointed out, those first 1,000 days, first 3 months of life are so critical for so many developmental processes. It's the immune system, it's the gut, it's the brain, and it's really a critical window. And like even in asthma, for example, you can't reverse it if you blow through those first 3 months, then try and fix it after that. It's too late because you've passed the developmental milestones that have to be affected. So my guess is that I don't know whether you'll be able to, I doubt if you'll be able to completely reverse all the brain stuff if you have the damage and then you try and correct it later, because you've missed the developmental program milestones you need to do at that time.
Thank you, Dr. Finlay. And there is a question on the NAFLD phenotype. in the malnourished mice with environmental enteropathy, and what is the cause, given that they don't have metabolic syndrome, right? They don't have, they actually have less fat intake than the control.
Paula, you want to handle that one? You had a lot to do with this. It's gonna be good for you.
Definitely. So it's interesting because we kind of have to step away from looking at metabolic disease as just in overnourished. We're actually seeing it in undernourished. And Dr. Jonathan Swan, lab in the UK, you just go and look up some of his work, and they're looking at the metabolic dysfunction that happens in undernourished children. And it's huge. And so what we're thinking is happening, talking about this early developmental window. So if you think about what's developing, so the body kind of makes certain decisions during development. Said, okay, I'm not going to have certain types of nutrients. And so what I need to do is hoard everything and store. So what we think is happening in the malnourished phenotype is that it's storing all these fat for a later time when it won't have it. And then that deposit is then leading to this NAFLD-like feature or phenotype that we see in as a malnutrition. And there's lots of works that have been done looking at predictive adaptive response, looking at what we call a thrifty gene or thrifty phenotype, where the body just kind of stores up things for later. And so that possibly is what's happening and why we see this deposit that's happening so early in life. But there's a lot of work looking at metabolic changes or dysfunction in undernutrition. And so I would really encourage everyone to kind of broaden our horizon a little bit, to just move away from just overnutrition being the one with the metabolic phenotype, and look at also undernutrition as having the same.
And Paula's favorite idea is what she calls the double burden of malnutrition, right? So initially, undernourished and overnourished. Exactly. And I think what surprised us the most was that, the phenotype of the metabolic disease we're seeing obesity is very similar to what we're seeing in these undernourished things. So when you talk about malnutrition, it's both over and under, and it's kind of both other sides of the coin, but it's the same coin type thing. And I think that really surprised. So we didn't expect to see this.
Yeah, definitely. And that was a part of my another model that I had, looking at the double burden of malnutrition that's happening in these LMICs. And so what we're seeing is that as countries begin to move into what we call the nutrition transition, so they're beginning to adopt our westernized diet and lifestyle, that we're seeing this increase in type 2 diabetes, obesity, and these metabolic diseases, but they're happening 30% faster, and they're at an earlier onset than we've seen in westernized culture, suggesting that this first hit of malnutrition actually programs something that is then causing this, when they then become exposed to this high fat diet, that they respond very differently. And so we're seeing disease manifest much earlier. So it definitely is a lot that's going on in those first windows that we need to pay attention to.
Yeah, thank you. Thank you for these answers. So we have now a question about current clinical practices in Canada. In many neonatal units in Canada, oral immune therapy with fresh human milk is a common practice for very preterm infants, and antibiotic use is very common. So any thoughts on how this may impact the antibiotic resistome?
Yeah, I think it is something that we definitely need to look at, because it's something that we just do without much thought to the microbiome. Clinicians are not really trained to look at the microbiome. They're not microbiologists, and so they don't really think about that. And their focus is very different. They're trying to save a life, right? And so we need to begin to look at how can we protect the microbiome in these very young children, so that we don't see what Bert was talking about, this asthma and all these different metabolic phenotypes. We see increase in childhood obesity when the microbiome is perturbed early in life. So there's so many factors that are related to that. And so we want to have some other methods that we can use and reduce the amount of antibiotics that we're using, or also to think of ways that we can protect the microbiome in the child. So maybe use of probiotics, maybe we can start studying some of those. Fecal transplant, maybe at some point, if that's safe for the infant, to be able to look at that, or using postbiotics, which is just extracting all the really good stuff that we want from the probiotics, and then administering those to children, maybe at the same time or prior to a dose of antibiotics. Correct? Any thoughts?
Yeah, I would add, I think when you think about antibiotics, everyone worries about AMR, right? This is the big thing coming. I would argue that actually the damage that antibiotics do to the microbiome is as big, if not bigger, in terms of human health. I mean, there's so many studies showing how rearranged microbiomes have major effects on obesity, diabetes, asthma, ADD. I mean, you name these things. So I think when we're talking about antibiotics, traditionally you're told, well, it might be a viral infection, but it can't hurt, right? It's just an antibiotic, right? That needs a major rethink that, yes, use antibiotics with life-threatening disease, but prophylactic, just like when we're using as growth supplements, that's got to be stopped, not just for AMR, but also for the damage it's causing to the microbiome. And again, anything you do in the microbiome, that first window of life, it is so critical. I mean, just anything, you know, like even born by C-section versus vaginal delivery, you have a 25% higher rate of getting asthma just by how you had it, because you didn't get the right microbiome. Same with breastfeeding, same with having a pet in your house. You drop asthma by 25%. Living on a farm decreases by 25%. So that first window is so critical and so touchy that you want to try and do your very darndest not to screw it up, because it impacts you the rest of your life.
Thank you. Dr. Putanen, I'm sorry I missed your raised hand earlier. Would you like to ask your question live?
Thank you. I just read it in the chat as well. That I missed the very beginning. I'm so sorry. I had a conflict. So I didn't see everything, but I caught Dr. Littlejohn, your presentation. Really exciting to see what you've shown with regard to malnutrition and the induction of the stress response in terms of resistance. And you really wonder the association and looking at globally the problem of antimicrobial resistance. It's often discussed, well, we need to have clean water, we need to have sanitation in the regions, and that's actually one of the drivers. But in fact, one of the drivers may well be is the malnutrition. Even if you don't fix the clean water and the sanitation, you may not have the resistance, the resistome, so to speak, being shared if we fixed malnutrition. You know, it does speak to this, you know, what's the primary driver in the global problem of resistance? And it may well, you know, in terms of where should we be focusing our efforts? Our focus on reduction of antimicrobials obviously is helpful, but the primary driver will very well may be partly to do with malnutrition and malnourishment throughout the world, which is exciting to think of that potentially being something that one can fix. Not easily, but something that one can fix.
Exactly. Yeah. And yeah, definitely. I agree. And not easily, but it is something that we should target. And I think we've ignored it. And this is why I'm really excited about this paper getting published, because I think it's a conversation that we need to have at the global level to begin. And I have reached out to the Micronutrient Nutrition Forum to actually start this conversation, because we do need to talk about it. We do need to talk about the risk that being malnourished has on antibiotic resistance. It's not just, and even on what we talked about earlier, just metabolic disease later in life. So it's not just looking right now at, oh, we're just using antibiotics. It's everything. And this is something that we have ignored. I don't think when you look at, it was very difficult for me when I was doing my research, looking at for nutrition papers related to antibiotic resistance. It's virtually none. The only groups that study any kind of like metals are environmentalists. And so they're way ahead of than us in the microbiology field. And so I've learned a lot from them, and they really take it seriously, understanding the relationship between metals and antibiotic resistance. And I think we need to as well.
Well, and looking at the literature, what's being, oh, sorry, Brett, but just looking at the literature in terms of looking at some of the associations, people infer that they're less healthy, they're more likely to get infection, they're more likely to be on antibiotics, and that's why they're getting this resistance. But your story is very different, which is
starting out.
Yeah.
Just to show you how complex this is, though, we had a cohort study in Ecuador looking at early life microbiome and asthma, and we looked at the risk factors. Of course, antibiotics increase the asthma rates and things, but ironically, the number one most at-risk thing you could do in terms of getting asthma in Ecuador was have clean water, because you weren't getting the worms and the things that they normally have, right? So these are very complex issues, and but antibiotics is one thing we can definitely control, and I think that, you know, the less we use, the better, in terms of, unless you really need them, because they are. I mean, there are several studies being shown that if you just treat kids with antibiotics, that doesn't actually, because the idea, while they're always being exposed to pathogens, let's just give them antibiotics all the time, no pathogens, they'll be fine, and that actually shows that does not work at all. All it does is increase resistance. So,
beautiful work. Thank you both.
Yeah, it shows a lack of understanding of antibiotics. Like if you just give more, you're just going to get more bacteria that you don't kill that are just going to develop resistance. So yeah.
So speaking of geographical differences, what about children living in North America? Do they have deficiencies that may be affecting their long-term health?
Yeah, there are some reports that I saw too, even in Europe. Vitamin E, vitamin C, vitamin D, magnesium. Most of us are deficient in magnesium. Selenium, like when was the last time anyone has gone to their doctor and they were like, let me check your micronutrient levels? Like we just don't think about it because we assume that we're all eating healthy, but we're not. We still have food deserts. Our our diets are depleted of the micronutrients by the time they get to us. Our guts are a mess. And so we have permeability. So there's malabsorption that's happening. And so we're just not getting the right nutrients. And if you look at even in the US, 51% of Americans have like a chronic disease, and chronic disease usually have like a low micronutrient deficiency. Sorry, have micronutrient deficiency accompanied with that. And so you have, this dance that's happening. And so we just don't look at it. So children in like Canada as well are at risk for, it's not maybe all 5, but at least 2 or 3 will definitely be there. But we just don't check.
Yeah, thank you. In the microbiota sequencing studies, did you look at the phage composition, and keeping in mind HGT, is phage therapy a promising approach?
So I did look at the phages in the low micronutrient group, and I did see a more bacteriophage that. Phage therapy is not an area of expertise for me, so I wouldn't be able to speak too well on that. But I know someone, a former postdoc in our lab, was tinkling a little bit with some of that back home in Israel. So I think some people are looking at it, but it's not an area that I'm most familiar with.
So we are jumping across many different topics. So this one is about microbiota and age Z-scores, especially in the stunting studies. Did you look at those? And if you did, what did you discover?
Yeah, the French group did. Pascal looked very closely at that. And yes, indeed, there's definitely differences. You can definitely have a characteristic stunted microbiome versus normal microbiome. I think the real question is, well, what does that mean? Like, why is that? Is it chicken and egg? Is it because they're stunted, you have different microbes, or you have different microbes that's causing the stunting? And I still say that if you'd ask me, how do the microbes actually cause stunting? We don't have an answer in that sense. So it's in the papers. There's 2 PNAS papers we published describing all this, and where they actually go into that, and they definitely see microbiome compositions associated with this, but taking it to the next level, what does that mean now is difficult till we go into animal studies.
Yeah, and there's definitely been some calculating a microbiome for age Z-score, but you definitely need, not something I think is possible right now with mice. Definitely we need hundreds of mice, which I wasn't going to do, but in children it is possible, and that has been calculated, and you can use that model to look at microbiome age in other cohorts. But like breast milk.
Yeah, so there's a question about potential markers of earlier or higher exposure to the fecal microbiota. Is this something that could be detected maybe with an oral test in children?
They're working on an oral test for their microbes. Is that precursor to potential stunting? So they're working on that in the Afrobiota study now. I think, you saw the pictures that I showed of the environments these kids are living in, that it's continual exposure, and how you control that, other than, hygiene, sanitation, and that whole thing. And they're looking at using the fecal samples, looking at the oral pharyngeal contamination, those things as a potential readout to how, but by that time, they're probably already well on the pathway to this. So I think you just look at where they live and say, yep, that's an at-risk area. I don't think you need a specific test. Paula, what do you say?
No, I agree. I don't know if there's any tests right now that we can administer to predict who will end up in a certain place.
Yeah, I think the effort really should be trying to reverse it. How do we actually prevent it or decrease it as fast as we can early in life? And, sanitation is one thing, but there was a big study that showed that that's not perfect. Even if you clean things up, it doesn't fix it. Well, why not? What's going on here? Do they just have resident microbes that are different, already programmed to this? So we're really, I think, struggling right now to come up with the magic wand that I think obviously Paul has shown that they better have strong micronutrient supplementation. There are sprinkles and things that people have been using to enhance this. They need a clean environment, avoid antibiotics for the damage. But then again, maybe that sometimes some studies shown that a dose of antibiotics actually starts in the right direction, cleans things out, allows you to reset and get going again. So more questions than answers.
Yeah, and so let's take the last question, which is more of a general one. It's about fecal transplantation, and what are your thoughts on fecal transplantation for fixing the gut microbiome?
Well, there's a lot of work in this area. So first of all, I say, despite the grossness, I think fecal transfers have been a fantastic proof of principle for proving that microbes can impact in life-threatening diseases. We've seen this really strongly in the C. difficile work, where you can basically cure a life-threatening disease that's failed in antibiotics. There's also some really nice studies coming out in inflammatory bowel disease, also colitis, where it's looking very promising. There's some studies coming out, but I would argue that, this is a human body fluid transfer, and we really don't know what all we're transferring. One of the adverse effects when they had two sisters, one was obese and the other was thin, had IBD. When they put the healthy obese fecal microbiota into the IBD twin sister, they cured the IBD, but she also gained weight, became obese, and so there's risk. So I think where the field is going is fecal transfer is a great proof of concept, but we have to get into administering collections of microbes they call live biotherapeutic products. And there's several companies. I'm involved in one that's actually doing this now, where they can put in a defined group of microbes for a defined biochemical reaction for a defined medical condition and actually show that works. Fecal transfers, it really depends on the donor, it depends on the disease, depends on the time of the day. There's one guy that this company uses. He's over in Europe. He lives in Belgium, and they basically train him all over Europe because he's got the magic feces. I don't know what it is, but this person.
I think we lost him. Is he frozen for you as well, Alina?
Sorry, I was on mute. Yes, I cannot hear Dr. Finlay either.
Yeah. Oh, okay.
It may be reconnecting.
Oh, there he is. He's back.
I came into UBC to hopefully have a good connection, internet connection. He says, your internet connection is weak. Too many students here. Sorry.
Yeah. Well, thank you. We're happy that you made it back. Okay.
Yeah, and Alina, I just want to add one comment to that. Recently, one of the things that I suggested that FMT should be considered the same as an organ transplant, because we're transplanting a virtual organ, and so the same way that we look at when we're putting in a liver, like, is it going to be a match? What's going to happen with the environment, the immune cells, everything that's around the environment. We should be doing the same with the microbiome. And then one of my concerns recently, obviously, now is the resistome. What are we actually transferring in? And how can that affect the recipient host? So I think there's a lot, like Chris saying, that the work that needs to be done, and we need to not all. One of the things is the society just gets very excited, and they're like, oh, FMT, whatever it is, probiotics, they get really excited. And like Brett said, they're always years ahead of the science to support it, and they usually just jump on the bandwagon, and then everyone starts selling everything like this is the magic cure, but the science isn't there yet. So be critical, read read science papers, and really get your information from credible sources, and not just people who are actually trying to sell you a product.
And do not DIY fecal transfers. You could break your intestine and die of sepsis. This is not something you do at home, despite what YouTube says you can do.
Thank you for your comments. Thank you for your talks and for the food for thoughts. Like, now we have many ideas and on future perspectives on where this field is going into. So thank you for being with us, and thank you for your time, all your time answering these many questions. It's very much appreciated. And hope to see you at our next seminars next year. Thank you also to all of our attendees for asking wonderful questions and being with us. Thank you, Chelsea, again, and to the Lawson family as well. Thank you.
Thank you so much.